is what attaches to these groups. So what amino acid goes in these R groups will determine the pH or make the amino acid acidic, basic, or neutral. Okay, any questions on that? So if I were you, on past exams, this is what he has drawn out, except for instead of keeping it this R, you're gonna see this big giant amino acid. Okay, you don't necessarily have to know what the amino acids are, but I have seen him put a big skeletal structure as following, and then say, please circle the amino acids. And I've had students that circled the carboxylic component or the nitrogen component, but instead of these. So just know these are where the amino acids are, okay? Okay, so let's draw some amino acids. I'm gonna put them in sort of like basic, neutral, and things like that. So let's see. See the best ones. I only know like 5 of them. Okay, cool. So here we go. Clear that up, clear that up. Sorry for those that we just started drawing late. Apologize. Okay, so in the pH, our body is, what is the pH of the body?
Is it 7.4?
Yeah, so normal body or physiological pH is 7.4. Okay. The question is, draw the full chemical structure of 5 amino acids. 5 amino acids. So how we can do that, we have to look at them, and I like looking at them in terms of the groups. So let's start with electrically charged side chains. So that means anything that has a charge. And we'll do the first one we can talk about is arginine, and I'm going to just abbreviate this as ARG. Okay, so arginine, and it looks like this. You have, what group is this that I'm drawing? Let's just keep studying as we go. What functional group is that?
Carboxylic acid.
Good. So what terminus is that?
C terminus.
C term. Good. Okay. And I know it sounds mundane to keep doing that, but it's really important that we are able to identify these. Okay. So then we have this. And now we have this. So this right here is this is going to be positively charged. So this is the structure of arginine. Okay. So when we're going through and drawing these, you'd have to kind of figure out, this is going to join on some sort of chain. So this is the R group. Okay. This is an R group. And some amino acids have these N and C terminuses so that we can keep adding and adding and adding and build these long chains, because when we think about DNA, RNA, everything is just like mixed in, right? It just keeps, each one of them just keeps stacking and stacking and stacking. That's what builds the amino acid. They're the building blocks, right, to proteins. So they're just kind of all stacked together. So we'll just keep doing this, but this is the structure. So if we're let's talk about pH. And we know that the scale is 1 to 14. So if we are positively charged, right, do you think we're basic, acidic, or what? If we have a positive charge, what does that mean in terms of, you know, what happened to the proton?
It's protonated.
So we're saying that this has been protonated. So what are we assuming that is this going to be an acidic or a basic amino acid?
Acidic.
Acidic, okay? Because if we look at this just in terms of my favorite, this, right? If I deprotonate this, then I'm going to have this. But if I protonate it, then I'm going to have this. Okay, so it's really nice sometimes if you just like to draw this, but then if I deprotonate, what am I?
Basic.
Minus.
Okay, and then I would be here as the conjugate acid, right? So I like to go back to this kind of thing because it kind of helps me figure out what it is that I am and what it is in terms of losing that H. So if I have a proton, if I'm protonated, another person. If I'm protonated, then I should be what? More acidic? Okay, so that's how I want you to start seeing things. Look at these charges and decide for yourself whether or not this is going to be an acidic, basic, or neutral. So this is arginine, so you can draw that. Another one we like to do is, oops.
Hey Liz, really quick.
Yes.
So that double bond R, is that connected to the N on that NH2?
No, this is just telling you that it would be an R group.
Oh,
You
okay.
can see this as an R group on a peptide bond.
Okay.
Okay, so all these that I draw would be the amino acid that would go into the R group of a normal peptide chain. Okay, so now we'll have histidine. Good question. So histidine is just HIS, and we'll leave it at that because I don't really think you need to know more than that. And we'll have, it's the same thing. Here we go, HO, and then we'll have this double bonded O, and then we're gonna draw this, and we'll have NH2, and then we'll have, and then this really wonky thing going on here. Okay. So what do we notice about this particular molecule? Does it have a charge? So we said that this one, what is this? Does this have a charge? No, we don't see any, I didn't draw in any charges. This has the positive charge. This doesn't have any charge. So this is a neutral amino acid, right? So histidine makes up a lot of our proteins. We use histidine for everything and it's neutral. So if, as far as this question goes, draw the full chemical structure for 5 amino acids showing the protonation state as they exist in the body, this is how it exists in the body. There is not a charge on this because if our body had charges all the time, that would mean that a lot of things are interacting, breaking down, right? So this is more of a neutral amino acid. There's no overall charge for this. That doesn't mean that it can't react with things, but in general, this is a neutral molecule. So I would keep an eye, I would know that one. It's really common. And yeah, this is positive. But the thing here too, though, is what is weird about this guy right here? What do we notice here? It has 1, 2, 3 bonds, right? Should it have, that's only how many electrons?
6,
Right? And so how much does he like to make?
8.
Right? So we can assume that this is here. So even though we're saying this is neutral, or as most would say, it's positive, but it's really not, this is questionable. You have to remember that there's still this lone pair here. Okay. And so it could still interact with water. Okay. So just because it has this lone pair here, we consider it still positive just because if this is a minus over here, and then we have an OH up here, right? We're assuming that that's negative. We would think, okay, well, Liz, this is overall negative, but then we have this guy over here. Okay, so these things will balance out and make it and change the way. This one right here is very important in our chemistry. Okay, has a lot of reactivity sites. This site's not going to do so much, but these ends are the ones that are going to do most of the interaction. Oh, what element is the low pair on? Nitrogen. Nitrogen is a lot of histidine. Histidine is built of nitrogen. And I know it's really hard for me to draw on this. Nitrogens. Okay. But when you're doing your homework for the virtual lab, you don't have to draw on these charges like this because in general, this does not show, it doesn't show those. So you can just leave it just like the first way I drew it. I'm just letting you know that there is potential for interaction there. Let's go through another one. It says that NH3 plus is basic. Why is that? So why would NH3 plus be basic? Why would this be basic? Because of this guy, right? Nitrogen, do you think he's going to be willing? And it says NH2, exactly. So nitrogen is a very special case. Remember when we went through and did talked about strong and weak acids and bases, we noticed that there was a lot of nitrogen in some of them, and those ones, these are not going to dissociate, right? So we have to differentiate between what makes something basic and acidic. When you're basic like this, what do you want to do? You're considered basic, but you have this charge. Your goal is that you're going to go and reach out for a proton. Okay, that's what I want you to think about when you look at NHs and nitrogens. These guys want to either interact or they're happy staying the same. So why they're basic is because they're not going to really charge. We don't see bases when we're doing acid-base chemistry. We don't really see the bases being the ones that are initiating the proton change. Bases are usually what? What do they do in return of, and what do they do in regards to hydrogen? Are they donating or are they accepting?
Accepting.
Right? So that's what we need to start thinking about is donating and accepting of protons. And on arginine is the plus on the NH2 on arginine. Yeah, this whole thing is the positive point. Yeah. So when we're thinking about nitrogen, these guys going to be doing donating or accepting?
Accepting.
That most of the time they're either going to do nothing at all and leave this positive charge until something strong enough can shred that and rip it off, right? So wouldn't arginine be basic? In a way, but what you need to know about is this overall charge. So that's why I'm trying to explain this versus this, because it's not the same in terms of pH, okay? Acids and bases don't necessarily equate to the acidity on a pH scale. Okay, so let's go ahead and think about a better way to get you guys to stop correlating pH with like proton source. And it's really hard to do that because on the last exam he gave you pH and you had to put it into Ka or pK or do some weird stuff. So let's just go ahead right now and clear out and really talk about pH, acid base, and then when I talk about acid base in terms of proton source. Okay, there's a difference. Because if I have a lot of proton in one, it just depends on what is doing the moving and because I can have a lot of protons in, and then we'll talk about, that kind of goes into Le Chatelier's principle about equilibrium. Even though I have a lot of proton, that means I can also make equal base. Like I can if I have a lot of proton in my reactants, I can make a lot of base in my products. Does that make sense? So it doesn't mean though that the overall pH could be neutral, but my product or my reactants could be super acidic and my products could be super basic, but then an acid and a base together would make a neutral. Is this kind of like making sense? I want you to think of it in terms of just individual groups. This overall amino acid though is what? You have one tiny little group that would be basic, but overall, what is this amino acid? And that's what you need to think about because we have this minus here, we have a plus here. So what is this overall amino acid, right? Is it kind of clicking now? So yeah, this might be positive and basic, and but is this overall thing an acidic or basic amino acid? That's the only way you'll know that is given by the pK of these amino acids, which you won't need to know. Okay, does that help a little bit? So let's go ahead and start with understanding. I want to pull up my old chart. Amino acids on the last exam because it's easier to, I get sidetracked with amino acids. So let's do here we go. Let's go ahead and clear this. If you guys have questions, you can always ask too. So just let me know. Okay. Okay. So right on. Let's go ahead and do this. let's just list out things, right, by, let's go do things by acidic. So these are overall acidic amino acids. We have this. And now this just lets you know, I draw things by their spatial orientation. You don't have to do this if you don't want to, but technically it's more accurate if you did do it. Also, I'm not drawing this all the way. I'm doing it condensed. We know what a carboxylic acid looks like, right? I'm just going to start drawing it like this, okay? Yes, arginine, those are amino acids, 100%. So this one right here is considered an acidic amino acid, and this is aspartic acid. So here's aspartic, okay? And that is known as this, and this is an amino acid, okay? Aspartic acid. Here's another one. We have glutamate, or glutamic acid, and it looks like this. NH3 positive, COO minus. Okay. We have to see though, remember when you start building in, what's the difference? What was the difference between this one and what's the difference between this one? What do we know is, what are the differences here? It's whatever's right here. There's an extra chain. Okay, so when we start building these amino acids, you just have to watch, well, these all have the same terminuses as they should. They should all have the same terminuses and the same bonds. Oh my gosh. All right. Oh yeah, that's different. I don't care. Math students. So this is glutamate, or glue. And it's known as this. Okay, these are the only ones that you should really know for acidic amino acids. Overall acidic amino acids, these are the only ones. This is it. So if we're acidic, we're assuming what? What are we assuming if you're overall, now I'm talking in terms of pH, I've already determined that my molecule has a lot of what? If I'm acidic, am I gonna have a lot of protons or a lot of protons or a lot of, or am I gonna have less protons?
A lot of protons.
Okay, so I have to have a good amount of proton. So proton concentration has to be high. Do we see that in this case with these guys? Even though we've said that these are basic, right? They're basic because that's what's holding the bonds together. Because what is this NH3? What is the peptide bond look like that we were talking about? You know, what is it that it looked like? We have to think about what a peptide bond is. I want you to be able to differentiate because he'll draw structures like this, you guys, and they'll be super condensed or thin, and you're going to say, oh my God, how is this any different than that last peptide bond that we drew? And they're not different. It's just they're just organized differently, I should say. So remember, the peptide bond has two components. Then the end term, I'm just going to call it that. So you have this. NH group, okay, some R group, okay, and then your double bonded O, OH, okay, that's one plus H2N, double bonded O, running out of room. Oh God, hold on, sorry guys. I don't know, there we go. Okay, and then you have this double bond comes up like this. You'll have up here, we should have some R group. There's an R group here. There's an R group here, this, and then we'll have this. Okay, so are we seeing how this and this are coming together? What's happening is, where's the reaction occurring? It's occurring between this and this, right? So that's where this is coming from.
And then that's the peptide bond, right?
And this will form the peptide bond. So you always will have one amino acid coming together to build that, Okay, so it just depends on, it's kind of hard to put these. So we'll be drawing structures of amino acids, but remember, amino acids are the building blocks to proteins, right? They're the subunits of proteins. So we're trying to think, how can I connect these individual amino acids together? And that answer is through the peptide bond. And that peptide bond happens when I cut this. And the byproduct is what? What happens if I cut these things? What am I losing? What kind of reaction do you think is occurring to a peptide bond?
Dehydration synthesis.
So, and that means to do what?
Water is the byproduct.
Water is the byproduct. And we can see that because I have one H here and I have two H's here. So we're trying to go ahead and make that byproduct. This is going to be the byproduct of a peptide bond between amino acids. Cool. As long as you fundamentally know that, you'll be 100% for the test. Let's go ahead, and while I'm at it, I told you I would pull up a quiz that's a test that's hidden, and we can actually do that too while we practice and keep drawing. Okay. Okay. But it's not really. Let's do this. Okay. Sorry, dudes. Let's go get this. There we go. Okay. All right. Salt bridges were kind of touched on. I think I saw something like that on a quiz or something. That's going to come back again too, because that forms. That's a lot of things. That deals a lot with some bonds too, because there could be S molecules, sulfur or phosphos in these amino acids. So we need to make sure you understand what a salt bridge. How are we supposed to put a drawing in a PDF? Okay, what I would recommend is just take a picture with your phone and then email that to yourself and either take a screenshot and put that into a Word doc, or that's my recommendation. I would just take a picture. Is the whole molecule just drawn as an amino acid? Is the whole molecule just drawn as an amino acid? So this whole molecule, yes, is the amino acid, but I'm showing you how we would connect. These would be two amino acids, right? I didn't draw what the R group was that would differentiate. It could be something that looks like this. It could be that something that looks like an R group could be like the histidine, right? So you have HN, N, right? And that could be the R group on top of this. Yeah, place a scan through your notes, yep, and convert everything to a PDF. I've seen people use old school and just take pictures. Okay, let's keep drawing. So those ones are straight up acidic. So if you were going to include those on your homework assignment and you knew that those were acidic, you could say, well, this is how it is, how you have to determine. I think he's just throwing people off draw, but the protonated state, you just have to show where on these things, you know, that would do the reacting. It's just what is going to be doing the reacting. Are these carboxylic acids going to be doing the reacting? No, they're just going to be connecting the different amino acids, but it is the amino acid itself that interacts with the body, not the backbone. Okay, the backbone is all of this stuff. This NH-OO bonded O, whatever's in the R group like this would be attached up in this R group because this is glutamate. Okay, and it would be here, and because we've got these positive and negative charges, that's what's going to interact with your cells. Is that clear? So on the peptide bond, which is multiple amino acids, we have other side chains. Those side chains will be doing the interacting, not the backbone. Okay, not the actual backbone of that. Cool. So let's go ahead and clear this, and let's keep drawing some amino acids. Let's draw another one that he likes. I don't want to make you draw horribly long ones. So, okay. something, okay. I also want to make this group too. If you have an acidic amino acid, I'll just flip it like this. The R group equals a carboxylic acid. It donates H plus, and because it is negatively charged. Okay, that's how this becomes an acidic amino acid. It's backwards from what we've been dealing with, okay? But it's still going to donate because carboxylic acid is this, right? So this is what's doing the donating, because that's going to get stripped off and make something else. Okay, so it's not the same as when we're dealing with, like, you have to think about it now different, even a little bit bigger, comes a little backwards when we're dealing with what makes something negative, because we would think positive, positively charged, that means you've been protonated, but that, remember, is just a conjugate acid, okay? It's a conjugate. That doesn't mean it is the acid. Okay, so even though it's fun to see how the protons move and it's good to understand how they move, you need to be aware that just because I'm a conjugate doesn't actually mean that I am the acid, right? Are we clear on that? Just because you're the conjugate acid doesn't mean you are the acid. That's just what's going to happen to when you're determining like which way a reaction will go. You look at the conjugates. Okay, so as far as basic groups go, basic amino acids, AAs, the R group equals the amine, and I'm going to put like this, sorry I'm writing in red, just look for the N or NH, okay, that's the amine component. It will accept H plus, and it is usually positively charged. And I want you to know that histidine, because we talked about histidine, so histidine ionizes at a pH of 6.0. Okay, so I want you to know this, because this is important biologically. Histidine makes up almost all of our, most of all of the interactions of drugs and things like that. We use histidine. And so I want you to see this pH because it's very close to our biological pH, which is why it makes up everything. So if I were you, hint, hint, on one of them, on at least for this assignment, I'd be drawing a histidine because it's so close to our biological pH. Okay. Can you explain what you mean by ionizes? Yes, I can. What I mean by ionizes is you'll have something, let's just use sodium chloride, which is simple. When something ionizes, these two things are going to be in solution separately. Ionization is just the coming of them together, right? So they ionize to form something. It's just a terminology we use for like coming together. Their ions meet. Okay. Okay, so those are just overall when you're looking and determining, look at the R groups. R groups, if it has a carboxylic acid, we know that it's an acidic amino acid, okay? If it's basic, we know that it'll have some sort of amine group, okay? So like I said, let's erase this. Hopefully everyone has that. I'm recording though. I'm gonna put black to black. So for instance, acidic, we already drew this. Here we go. This is how I would draw every amino acid that I draw has the same backbone right here. Carboxylic acid, there's the H3, there's our amine part, peptide bond. So that's one start of amino acid backbone. Here's the start of another amino acid backbone. It's all the same. What changes is what goes here. And I'll draw that in green. So this backbone is consistent of this. And this backbone has this. Okay, this is the R group. This is the R group. So when you're drawing amino acids, I'm just going to do R1, because remember these two can come together, right? When you're drawing amino acids, draw it this way. Start practicing it this way. Everything should start with this. Everything. That is how it should start. this part will change. And it's easier to see one subgroup, one subgroup, one subgroup. This H right here is what we'll do, what we can do some reacting with, but this is how everything will look. You'll draw every amino acid as follows. H3N plus to a carbon, has a hydrogen and some carboxylic acid. There we go. That's how we draw in our group. Connect these guys, okay? As far as which amino acid is that? So this is gonna be glutamate, and this is going to be aspartate. Okay, and these are acidic amino acids. And why? Because on our group, we have a carboxylic acid. Okay, so we have an acid here. Acid. So as far as acids and bases here goes, what would they be in like a body? Would they be like that in the body pH? So remember, our pH, so if we're an acid, what do you think our pH is going to be? It's going to be low, right? If this is physiological pH and then this is basic and this is acidic, these two would be around this, like, I would say between 5 and 6 pH. So that's where they would run as far as that. They're pretty close here, but if we walked around in our bodies with these minuses over here, what do you think is going to happen? There should be a lot of reactivity, right? So do we want, and if we have a lot of aspartate, do we drink things with aspartate, right? And that's why our body is able to use that, and that's why we get caffeine and sweet and all that thing, those things, because our body actually uses these charges and it can cross the blood-brain barrier. So yes, when there's some charges to these things, our bodies, they will respond to that and then fix it and try and cut this up and use it, or at least make it neutral so that we can get back to functions, normal biological functions. But you have to think about it. If it's smaller, this thing can travel. And so in the pH, in the body of the pH of the body, I would say it's around a 5 or a 6. Do the acids make them different from each other? Why do they have different names if they look the same? This has a whole new carbon chain. So what that means is when we have bigger carbon chains like this, if you kind of think about it in terms of size. So you could have aspartame, right? We've heard of aspartame. Aspartame is like this, but like glutamate is like this. And if we think about the blood-brain barrier and the spots for these, who do you think is going to be able to travel membranes faster or get through membranes better? Aspartame, or glutamate? Which one? This guy, right? So he's going to be able to get through the blood-brain barrier, but we don't actually have sugar, glucose, or any sort of that going to our brain, do we? Glucose is really big. So what happens is this guy has to have like other little helpers break this down. So it's important to recognize these structures because remember, size, shape, charge, all of these are talking about solubility, right? And what's the likelihood of this to be to actually go and be dissolved within the body. So yes, it's very important to know that this is going to have a harder time because we have these extra carbons. It's going to make it bigger and wonkier than just this, which has one carbon chain. So make sure you're aware of that. Could we draw those too, just for like that? Yeah, you can draw these just for like that, lab. Make sure they're acid and then just talk about this and why it makes it acid, makes it acidic. This is what makes it acidic, this R group. For sure. Okay, so let's keep going. Clear. Okay, another one that I like is something that he'll also want you to know is that those two that I just drew, they're also polar, right? We need to be able to differentiate polar side chains. We know what it means to be polar. So please note that on the exam, he'll ask you, is it a polar amino acid or non-polar amino acid? You already know this terminology. You already know it. And so you have this carboxylic acid. We know this is very electronegative, so we know that it's going to be polar. Okay, I haven't heard him ask about dipole-dipole moments, but just if it's a polar side chain or non-polar side chain. So I want you to be aware, start using that terminology again when you're going over amino acids. This would be an acidic, so aspartate or would be an acidic because of the carboxylic acid, and it would be polar, okay? So we know that it's going to want to interact or has the potential to interact, okay? Is that the only thing that affects polarity is the side chain? 100%. The 100%. We only look at the side chain when we're talking about these peptide bonds between amino acids. The R group will do everything and determine everything. R group will determine the acidity. Good question. Okay, basics. Here we go. Again, for the basic, we already drew histidine. So I'll draw it one more time for those that didn't get a chance to do it. Always H3N, okay? We have this. You can, and I'm gonna draw that together and then COO minus, okay? For the sake, I'm actually gonna, because I'm running out of room, I'll draw it this way. But it's the same way we were already drawing it, okay? Then we have, this one will have a CH2. And then histidine is known for this beautiful, like, pentane, kind of like looking this weird little doohickey. Don't draw it, it's terrible. Whatever you do. Okay, so it looks like this. This is our histidine, okay? So the other histidine that I drew, I just drew it as an N. Go ahead and add that H in right now because in most of his exams, he'll have it drawn like this. So this is how we determine. So what makes something basic are HNs. HN, and I'm just gonna put, I'll put, actually, let's undo that. Let's do it the right way. What makes something basic, a basic amino acid is this group, okay? This, okay? There you go, basics. Nitrogen, look at nitrogen. So here is histidine. Another one we could draw would be lysine. So lysine, I'm just gonna do like this because it's the same backbone. Okay, and the R group is just going to be a huge freaking chain of carbons. Okay, so that one is lysine. Okay, and let's put, this is LYS, lysine. And basic, these amino acids, let me ask you, are these polar or non-polar? Would these be polar or non-polar? Non-polar. So when you're saying non-polar, remember, non-polar is indicative that there's no, I'm not gonna write that, that there's no electronegative atom or atom available. Is there an electronegative atom still available on this molecule?
Yes.
What is it? Think of the periodic table.
Nitrogen.
Yes. Okay, a fundamental of life, these guys right here, CNOF. Okay, so it would be a polar molecule. Even though it's basic, it is polar. Okay, it still has an electronegative atom that is still either not, that's not happy or temporarily happy. Okay, so these basics are still amino acids that are polar. Okay, they're polar basic amino acids. Lysine, histidine, and then another one that is common is, I already wrote arginine. So it's the same exact thing, a huge big long carbon chain. And then here we'll have NH with this double bonded, oh, that's pretty trashy. Let's undo that. This, and then what has on here is another carbon double bonded to an NH. And that's 2 plus. This is 2 plus. And then down here is a single bonded NH2. Okay, so we have a lot of nitrogen down here. But overall, it is still a basic amino acid. And this is arginine, ARG. Okay, so that is something that we need to pay attention to. Is just look for in basic amino acids, you're going to notice a lot of nitrogens. Okay. In acidics, you're going to notice a lot of carboxylic acids. Those are the dead giveaways. That's all you really know. That's like that. I've given like 6, I think, so far. Neutral amino acids, we can go over. They're just really difficult to handle. So these are all what we like to call ionizable. So because they all have ions, these can all do some sort of reaction. So ionization, again, we were asked what the definition of is it's just something that has an ion of charge, something that has a charge. It's pretty much just a charged amino acid. And then you can just differentiate based on that R group if it's acidic or basic. So is it an ion? Yes, all these are ions. They have these little charges here that are not satiated. Non-ions amino acids would be this. It could be alanine. You can have leucine. You could have valine, isoleucine, proline. There's 20 amino acids, but let's just draw a few of these. So remember our same backbone. I'm not going to draw it. For alanine, that's gross. I don't want to draw that one. I guess all these have freaking cyclics in there. Okay. Let's see the best way to draw these ickies. Okay. 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 Alanine. Okay, he looks like this. So here's alanine. I'm going to draw them independently. H3N plus. We know this, right? Get used to drawing that. Remember, this could be drawn with the CO up here and the H there if you like to draw it that way. It's still the same thing. The interactive spot is this single carbon. Okay, so what comes off of that? So alanine, all he has is this. So if you look at alanine, if this is the R group, is he polar or nonpolar? Is he polar or nonpolar?
Nonpolar.
Why? What's the R group?
It's
just a hydrocarbon. Yep, this R group. Here is the backbone of the amino acid. This is the R group. He is a non-polar amino acid. Okay? He's a non-polar side chain. Look at that. Boring. Nothing happened in there. Okay? So what do you think that's going to do with our bodies and our physiological pH? Is it going to do anything? No. Okay? Nothing. It's going to do absolutely nothing. Leucine. Let's draw leucine. Here we go. H3N plus, CH, COO minus. There's our N terminus, C terminus. Here's my side chain. Leucine looks like this. CH2, CH, and then it's usually drawn like this, which is an H3C, a CH3, and over here is a CH what? 3. Okay, so if this is my R group, what are we finding a trend here? Is it polar or non-polar? It's non-polar. So do we think that this is a basic or an acidic or neutral amino acid? It's non-polar.
Neutral.
Neutral. Key trend with these non-polar side chains, they're all going to mostly be straight up carbons. Okay, the only one is tryptophan, and I'm not drawing that because he hasn't talked about it and he won't talk about it, but it does have a nitrogen group, but it's normal, it's stabilized, and it won't do anything because it's on a cyclic ring. Okay, and then another one is methionine. Methionine is like this, and so I would know methionine, and I would know this just on the back of your hand, of your just for your own work. So in case he doesn't try to, there's not an E here. So methionine and tryptophan, this is going to have an NH group, and this is going to have a sulfur group. This one that we just drew, the name of this one is leucine. and it's a non-polar neutral amino acid. Okay, non-polar, no charge. R group, no charge. These are still considered non-polar amino acids, but what makes them special is they have a sulfur and they have a nitrogen. I'm not going to draw them. You can Google them, but just know that if you were to say, you know, which I've seen it kind of like as like a tricky question, which one of these contains sulfur? And that was 2 exams ago. And he really pressed in class amino acids, and they had quite all of these ones. They had leucine, and all the ones that were hydrocarbons, and then he had methionine in there. They're all still nonpolar, but this has a sulfur. So you could go in depth about sulfur bridges, okay? Because that's how we'll be making sulfur bridges or salt bridges. So it's kind of nice to know which ones have sulfur in them. So you can do that, but let's focus on that as the next test gets closer. Okay, that's 2 more. And then let's just do serine. So serine is a good one. Serine, again, we have H3N plus, CH, COO minus, okay, traditional backbone. And I'll draw it. Side chain looks like this. We have CH2OH. Now, this is not carboxylic acid, okay? Just because we see OH on our functional group test, a lot of people were seeing OHs. This is an alcohol, okay? So this is still very reactive though. So in the sense, is this polar or non-polar? This part makes it what?
Polar?
Polar, okay? But this is not carboxylic acid. Please note, it is not this, okay? Acidic amino acids have carboxylic acids, not this carbon with an OH. This is not carboxylic acid. So please see the structures. These are not the same thing, okay? This is just an alcohol group, and it is polar, okay? It doesn't mean that it's acidic. Doesn't carboxylic acid have to have a carbonyl? So it's going to have this component, right? It has to have this part, yes. But when people draw their peptide bonds wrong, okay, they'll usually miss this carbon here and other things, and then they'll try and make it to look like this, so they associate. So make sure you're oh goodness, I did it again. Okay, it's gonna keep drawing. Oh crap, I can't fix that. How do I fix that? I don't want my pencil. Okay, so yeah, just let me draw this again. That is not the same as a carboxylic acid, okay? Please, please, please. He likes to do that on tests. And it's just an alcohol here, okay? Cool, so this is serine. And he is a polar amino acid. Cool. I just want to let you know that you might, so I would say that there's 4 groups, just to reiterate, 4 groups, amino acids. So are neutral amino acids polar? They can be either polar or non-polar. Because neutral doesn't necessarily mean like no charge. Because if you look at the overall charge of like an amino acid, you have positives, negatives, and then this OH, but it can still kind of work. Just because you have it like this and you see this alcohol group, you would assume it's like this, but still it doesn't mean that it's a negatively charged amino acid. This is still considered like just a neutral amino acid. It's not strong enough to do a lot of work here, because we know that this, even though this is a strong base in terms of amino acids, well, a strong base is this, right? In terms of amino acids, this is still relatively a bad leaving group. So OH is a bad leaving group. He's not going to willingly just fly off of an amino acid or an R group, a chain. OH is not going to be like, bye, peace out, I'm going to go find something to do. He's too weak in terms of a big group like that, a big molecule. So we don't really necessarily say we would say that they're most of the time neutral. Okay, it's not strong enough. All right, so 4 groups of amino acids, I'll tell you right here. Nonpolar, polar, and then electrically charged. And the reason I say 4 is because I consider this like a mediocre group, because you have acidic and basic. And I focus more on these as the 3 and 4. So 1, 2, 3, 4. But this is just what they're technically called. Okay. So if you hear him saying which one of these is electrically charged, you automatically should be knowing you're dealing with something that is straight up acidic or basic. Okay. So please note that. Otherwise, you're just going to have these types of amino acids. And for the most part, they are relatively neutral because they can't kind of interact without help. So they couldn't interact without something else helping them, either other amino acids or whatever chemical or molecule you're trying to make. Okay. In addition, so usually I say that because of drugs, right? Drugs affect our proteins, and so it depends on the drug and the receptors of the drug and the amino acids that would kind of change things. But that is really into biochem, and we don't need to go there. You just need to know this stuff fundamentally. Okay, what else? I think that's pretty much it for that. I think I gave a lot of amino acid drawings. Let's see. Does anybody have any questions, comments, or concerns on this lab? The amino acids labs. I should actually show you, see what they did for the last test. So nobody has any questions at all? What is the homework doing? Who's doing the homework? What is the homework on? Is it drawing amino acids? So like this, let me just go back really fast, quick change of thought. One of his questions was, what type of amino acid is histidine? Polar, non-polar, acidic, or basic? What type of amino acid is histidine? We just went over that. Look at that R group. What's in that R group? So it does have, it is technically polar, right? But is it neutral, basic, acidic, or what?
It's neutral, right?
So remember, we have histidine, we drew it like this. And then you could I'll draw it the layman's way. So is this has a charge, so we know it has to be acidic or basic because it has this ion here. This is like an ion, right? Anything an ion is just what happens to this in terms of electrons. So it's positive here. So we have to determine if it's acidic or basic. Also, please look at these groups. What makes up histidine? Nitrogen. And what did we say makes an acidic versus a basic amino acid? Right? So don't let this fool you. This is technically polar, but is it neutral? No, it's not neutral because it has a charge. Okay, it has a charge. That's why we use histidine in a lot of things. Okay, it has a charge. Overall, histidine is a charged molecule. It has an NH and an NH plus. Okay, if this wasn't here and it was just 2 NHs, totally fine, but it doesn't. It lives with this positive charge here, therefore it is basic. Okay, so the question, that answer that was on his last test would be D, basic. It's a basic amino acid. Okay, so I would know histidine. I would know, and I'll, as soon as, like I said, as soon as this test gets closer, which it isn't yet, So a more positive charge means it's acidic and a more negative charge means it's basic. No, negative is acidic because you guys are still thinking of it in terms of conjugate acids and bases, right? Because we just got off of that exam. Acidic, we have more negatives. Okay, because what's going to happen is this can react with H. Okay, so acidics, more acidic amino acids are made up of carboxylic acids. Basics are amines. Okay, acidic, this is literally just straightforward. Acidic, carboxylic acids. Okay, so negatively charged, you're more acidic. Why? Because you are the acid. Okay, you don't have to think about it in terms of conjugates. Carboxylic acid, acidic, any sort of amine, NH, you are basic. Are all the ones you drew, how would they be in the body? So aspartate is acidic since it has a COH, correct. Aspartic acid, glutamate acid, okay? You'll hear things like that, and then otherwise it's just gonna be histidine, glutamine, lysine, okay? Yep, perfect. It has a COOH on the R group, it's acidic. If it has an NH on the R group, it is basic. Remember, that's why peptide bonds are zwitterions. You have the amine component, you have the carboxylic component, plus and a minus, zwitterion, two poles have a plus and a minus, and a molecule has both. Okay, this is why he mentioned zwitterions. Okay, yep. Are all the ones you drew, how would they be in the body pH? I think that this question is just kind of a little bit weird. All he wants to know is how would the amino acid you drew interact with the body to maintain a pH of 7.5. So if we had, for instance, this amino acid and it's in the body, what would our body have to do to get rid of this positive charge? Or how would it affect this positive charge? If we had a lot of histidine built up in our body, what do you think our body's pH is going to do from a 7.4? How is it going to change our body? If we had tons of histidine in our body, what's going to happen? Yes. And is that good for us? Is it good for us for our pH to be more basic? Yeah, so the pH will increase, right? No, it's not, because we want to maintain this. So what do we have to do to these amino acid groups? We either have to add more things to that backbone and add more amino acids to balance out this positive and negative charge, or we have to do something, have a biological function where we modify the histidine and protect it. So that's why amino acids, I'll just draw a really tacky picture. So as far as that question goes, it just says how, as they exist in the body, that is how it would exist in the body. Okay, but we have also would have it protected with interacting other molecules. Okay, so you don't just have free floating histidine. So if you had some sort of acidic, so let's draw this big chain, amino acids. And then I'm going to do like this, and I'm going to do like this, which is indicative that that's the end. So how does the side chain influence the properties of amino acid? How does the side chain influence? The side chain influences based on the polarity, non-polarity, or ionic charge, okay? If I am positively charged, I'm willing to give away this hydrogen so that can interact with maybe like bicarb, or that could interact with any sort of free-floating this in my body, because this can go and pick this up, and now I'd be left with this on the amino acid, okay? So what we can do is we can actually interact with these amino acids to create something different, okay? So now I've stripped that H. What histidine is good for is acting as a proton source, so that we could maybe go through ATP, or we could we could hydrolyze this, and we could utilize this proton for any sort of like proton pumps, Okay, so that's why it's important to have these things readily available. Also, if we have tons of this built up in our system, we would need a carboxylic acid so that that carboxylic acid can grab it and kind of remove this plus charge that's been taking up our body. So if we have a cell, let's clear all this. If we have a nice little cell that looks like this, okay, and this white is everywhere else. Here's the cell, everything else. If my cell has a lot of this, and it's built up a lot of positive charge inside, and then we have just protein blocks. These are my tacky amino acids, okay? And this whole thing makes up a protein. If I have these chains, right? This is really, really sorry. This is very like metaphor. Okay, if I have these chains like this on this is the protein, and we know proteins make up amino acids, and I have these side chains that are filled with this, right? What can I do? And my goal is, this is a lot of buildup here. What can happen is, we know proteins can cross membranes. We know that because there's these little channels and stuff, and we learn about this in biology. For as far as the chemical standpoint goes, we can have amino acids that enter cells via transporters or whatever and pick these up. So now what is my overall charge of the cell going to be? It went from super this to now what? Either neutral, hopefully it's neutral, which is a biological, we call that like some sort of equilibrium or homeostasis, right? We want some sort of homeostatic environment, or it could go to more negative, which we don't really want, right? So if we took too many H's, but the side chains influence that by helping manage this homeostatic balance for our cells and our bodies. Okay, that's why amino acids are very important. That's why proteins like DNA, RNA, proteins, that's why it's the central dogma of our life, because those proteins, the subunits, amino acids, that's what helps regulate that normal body pH. Without the help of the amino acids, we would have free-floating charges, either so much H or so much plus or so much minus, negative, and we wouldn't be able to regulate, and then all of our cells would suffer because of it. Okay, so these fundamental, these blocks, these building blocks, these amino acids help maintain this physiological homeostatic environment for our bodies, and that's why it can still work. Okay, that's why we also have protein pumps. That's why we have electron transport chain, okay, because we have to have somewhere from where these amino acids have created these vesicles, we have to have some place for these protons to go. So if they can't get bound to an amino acid, then they need to be gone, they need to go through other apparatuses, which is how our body changed and adapted. But for the most part, side chains help dictate the free-floating ions and help control the ions in the body as we build and grow and replicate. Okay, so that's why they're important. I wouldn't want to have a protein that's neutral going in and trying to take off hydrogens. It doesn't work that way, right? So that's why it's important to know when we're dealing with drug interactions, if we want it specifically to a target, something that is acidic or basic or has a proton or needs a proton, we can build a drug that way based on the polarity or the side chain, okay? So if we know that some sort of genetic disease has a lot of histidines, we know that then that is very basic. Our drug should target that basicity, those nitrogens, okay, and be able to neutralize. Does that kind of help? That's kind of like the overall takeaway from this kind of thing. Cool, and that's why I think he talked about COVID, because you know, everything is built with amino acids. So it's like, how can we target that with the drugs? How can we use certain drugs to help subside the symptoms, but based on the structure of COVID? So that's why it's important to know these side chains. Okay. Any other questions? Oh, somebody's coming in. Any other questions, comments, concerns about it, about this? This should be easy for you guys. Like I said, this is a very easy lab. Just get it done. It's just kind of time consuming because you have to watch videos. But you should be good. My next, so it's 2:13 now. So we'll cut this. We'll cut this. Even if you just entered, we're going to cut it. I recorded it. So if you missed it, you can go and look back. What do you guys need from me? When is your next test? Do we have that information? I didn't look. I think it was given, but let's see. So I can write stuff out. When is our next exam announcements? So there's not a fifth question. It's just like, yeah, I didn't see, I don't think so. I printed this out and I don't have a fifth question. I think you just hit enter. So I wouldn't be worried about it. I would just do 1 through 4. Yeah. Let me look again while I look on April 6th lab. Virtual lab. Yeah, I think he just hit enter. I think it's only 4 questions. So good question. The final is on the 25th. Okay. Do you guys have an exam 4 or that is the final, right? Super. Okay. So here's my thing. I think what he's going to be doing on the final is it's going to be cumulative. And I know that freaks people out. So we will do the same thing that we did last time, and we will hold multiple reviews focusing on smaller chunks, okay? Because that stuff with like Lewis structures and functional groups is going to come back to haunt you, okay? So you need to be familiar with that. There's going to be small amounts of acids and bases. Yep, it will be 20 points for 10, 11, and 70, exactly. So here's my tip for you. You guys just took acids, bases, and gas laws. If you are comfortable in that, I would take that and put that on the back burner. What I would focus now on is things like this. And I'm not going to go over review today, but I'll start giving you a list so it kind of helps you. You need to talk about hydrophobic effects. You need to be able to do Lewis structures. These are the things that are going to start. I don't know if 12 is on it. You need to talk about atom size. And I'm going to put in this periodic table or periodic trends. Okay. You need to know about half-lives. So, and how to calculate. Okay. You need to know about chemical formulas. And when I say chemical formulas, I mean correct chemical formulas. So this kind of goes with this, okay? You need to know, he might give you a bunch of molecules and you have to select the aqueous solution with the highest pH. So you'll have to do some hydration. Okay, this stuff is coming back. Hydration reactions, condensation. Okay, those kind of things ringing a bell. They're coming back. Polarity, nonpolar, polar, nonpolar molecules and identifying those. You need to talk about energy. And I'm going to put on here and graphs. So that kind of thing that dealt with, you know, endothermic, exothermic reactions. You need to know that kind of stuff. Okay. That's a horrible graph, but you get the gist. You need to talk about oxidation states. Okay, and this list just keeps going. Let's see. I don't think so. Yeah. There's going to be mass. So we already determined the mass. So mass on there. Hydronium concentration will be on there again. Volumes will be on there again. So M1V1 or C1V1. Talking about polarity, circle and naming the functional groups and balancing. Okay. We already put that on. Did I put functional groups on here? Well, functional groups will be on there. Okay. And talking about activation energy, enzymes. I'm not spelling things right. Okay, that's just a small list. So here's what I want from you guys. Today is Tuesday. I don't think we need to hold any reviews this week. Let's just freaking stop with the chemistry for a while. We'll reconvene next week. Throughout this week though and the weekend, I encourage you to go through this mini list here that we just made and figure out where your weak points are. Okay. When I mean your weak points, I mean the stuff you're like, oh my gosh, I don't even know if I have practice problems on that stuff. I want you to figure that out, because then what I can do is I like making efficient, concise study guides. If it's not something that we can't just like have a little conversation about, I don't want to have to put that on there. Okay. I don't mind doing more math problems, but if there's certain things that people are like, I really need help with, I'd rather build a study guide around those weak things than just making a general one. Because it doesn't help anybody if you get a study guide that's 6 pages long and you're like, oh, where do I begin, right? Those are stupid. Like this right here is causing me so much anxiety looking at it, because I'm thinking that's going to be 3 hours of review. Okay, so let's go through each one of you. There's like 15 here, at least still. Go through, figure out what your weakest ones are. Get me those weak points. Hey, Liz, I really need help with blah, blah. Hey, Liz, I really need help with blah, blah, blah. Hey, Liz, I'm actually pretty good with everything. I just want this, blah. Okay, do that. I will make a study guide. I will mass produce it. I will email you. Then we can work on that in chunks. Okay, we can start with this. Next review session, we'll do this, and we'll plan ahead properly. Because I think that works, since now he split the final. We have multiple, we can just kind of like condense these study sessions instead of one big massive one. Cool? Okay. Get your homeworks, get your labs in. We're still making sure those points. I'm not gonna lie to you, for those of you that are not in my lab, okay, please, I encourage you to please reach out to me once your lab grade gets put in. You should be, because we as TAs all put in your lab grade. So I know that my lab students are getting 100% unless I've had students blatantly not show up for the time that we were in class on campus, okay? As far as this stuff goes, everyone's receiving credit for my lab. If you're not in my lab, but you've been attending these sessions regularly, and I know I've noticed a few of those names, and you get your lab grade and it's not what you expected, reach out to me. Okay, because I can go in and say, hey, Tom, I didn't see TAs. TAs didn't hold online virtual labs. And I don't want you guys to be penalized. I know I'm not penalizing my students because they're putting in the effort, and I see effort from other students that weren't in my lab. So please, when that time comes and the semester is dwindling down, take a look at your lab grades. Okay, if your lab, they're out of 50 points. So if your lab grades are not what you think that they should be, and you felt that you've attended every lab up until you could, and you've attended these, I need you to reach out to me. Okay, so that I can advocate for you as a chem student. Okay, other than that, my labbies don't have anything to worry about. So you'll be good to go. Do not worry about those points. You're very welcome. But you just have to communicate with me, okay? If you have issues with quizzes or tests, okay, I had a student that just took the second half of the last test and her internet went out. Please email me, okay? Sometimes Tom doesn't respond relatively quick. I do. So email me, and we can we'll make some accommodations for that, because, but most people had no issues with their internet, but I do know that sometimes freaky things happen. Okay, so no judgments, just email me right away and be proactive about it. The more you communicate, the better off it is. So I have record, hey, this student, the exam was open until this point, he emailed me or she emailed me here. They emailed me here, let's reopen it or whatever. Okay, so just do that so we get your points. Please finish these assignments. Mastering chemistry. I don't really care about mastering chemistry. This is going to go on public record. I don't care about it. So if you're having troubles with the structure building, reach out to me, okay? But please include the problem number, because I can't go and fish around. So, hey, I really need help with 23A or 23.7 or whatever the heck mastering chemistry does, and I can go in and see what that is, screenshot it back to you. Okay, I'm relatively good. Just give me a couple hours to respond, because I do have quite a bit of emails, but I will get it to you. Okay, get your points, finish your homework. Please finish strong. I know it's annoying and the videos suck. keep attending, keep working on it. I will set you up for success. I have all of the quizzes and tests and past exams. We'll work on that. We'll go through reviews. You'll be good. Okay? Don't let this stuff scare you, but I would start now focusing in on this kind of stuff. Okay? He's used a lot of amino acid questions in the past, so we'll be covered there. But as far as the main review goes, like the big chunk of the next test, it's old stuff. So go through triage and figure out what you're weak at so we can focus on those things. Okay? Alrighty. I'm going to end this thing. Email me, but get some rest and do something other than chemistry this week, please. Okay? You guys did great on your exams. So, alrighty. You guys have a good night. Okay? Let's go ahead and stop this.